referred to as the triple threat, a clinician who can see patients, a teacher, pedagogue, and a researcher. And he does all three, and somewhere along the way, even though he tried to avoid it, he got roped into being chair of the department too. So he's now an administrator as well. But, I was saying before, my father, may rest in peace, used to have a saying that it's better to be lucky than smart. And Richard was lucky just by dint of timing, because he went into neurology just at the time that neurodegenerative diseases, and specifically Alzheimer's disease, were beginning to be appreciated. Because before that, and it's hard for young people to understand that, it was really sort of blended into aging. And he started his career research-wise focusing on neurodegenerative diseases, Parkinson's, Huntington's, even HIV with dementia, and what became Alzheimer's disease, and focusing on the neurochemistry, the pharmacology, the pathology. But then it was like the early 90s, and molecular genetics sort of changed the game. And he had either the foresight or the luck to observe the hereditary nature of dementia and Alzheimer's disease, and then effected collaborations with Jim Gusella and Peter St. George Hislop to pursue the molecular genetics, and that has grown into really an enormous research enterprise, which is housed in the medical school and university, but specifically in two institutes, the Taub and the Sergievsky Institute. So I'm not going to go on and talk about the numerous publications, but he really stayed current with the cutting edge of the science in the field on a disease that, like autism more recently, came into focus, and by dint of the demographic trends in our population, is now arguably, probably, along with cancer and what used to be heart disease, as the biggest public health challenges that medicine faces. Not surprisingly, his work has been recognized in a whole variety of ways with awards, but just to mention two, he was elected in 2001 to the National Academy of Medicine, and also as a fellow in the Association of the Advancement of Science. So please join me in welcoming Dr. Richard Mayeux.
Thank you. It's always a pleasure to come here. I often tell people that I owe a lot of the success to the department. I think I was one of the first neurologists to have an appointment in psychiatry, and now there's a bunch of us that have joint appointments. But part of it was a deal made with the then chairman, Herb Pardis, who, as he arrived here, wanted to, I was just starting my career working on Alzheimer's disease, and he wanted to get involved in Alzheimer's disease. So he said, what can I do? Let's not battle, let's not have a turf battle. What can I do to make it happen? And in the old Psychiatric Institute, there was a kitchen on the 13th floor, and we turned it into a clinic. And the idea was that I was interested in the population around Washington Heights, and we couldn't see them in the Neurological Institute because they had to pay. And I said we needed this population. We needed to study this population, and so, of course, nobody had to pay in the Psychiatric Institute. So that was the start. And then I had been recruited back from Boston to work with Nancy Wexler collecting DNA for the Huntington's Project, and in walked a family from the Dominican Republic with early onset Alzheimer's disease. And so I knew it was Alzheimer's disease, even though they had involuntary movements. But I collected the DNA anyway, and I sent it to Jim Gusella in Boston. And I called him immediately, and I said, look, this isn't Huntington's disease, it's Alzheimer's disease, but we should save the DNA anyway. And he said, oh, that's okay. I have a young postdoc named Peter St. George Hislop who happens to be interested in Alzheimer's disease. And that led to the first linkage paper in Alzheimer's disease, and eventually the cloning of the presenilin-1 gene. So my origins really are in this, my research origins anyway, really are joint effort, and I really appreciate all the connections I have to psychiatry. So I'm going to walk you through the Alzheimer's disease sequencing project. And this is largely due to the effort of Barack Obama, who realized that there are about 6 million people in the U.S. right now with Alzheimer's disease and about 15 million worldwide. And that's a misleading number, because that's the people that we know have Alzheimer's disease. They've seen someone, and someone's labeled them. In the community studies here and elsewhere, we're pretty certain that at least that many more are at home, and they're in a sort of a protected network, and nobody knows they have Alzheimer's disease. And we see this all the time in Washington Heights. So it's a real serious problem. And having spent the better part of 10 or 15 years looking at what people ate, what they do, and everything else, and understanding that there was no major environmental factor that we could find, we felt lucky that the genomic era was starting and then sort of evolved right under our noses, so we were able to take advantage of that. And what I'm going to talk to you about today is sort of a dream come true for me. We've been able to identify a series of genes, and I'm going to walk through that. But now we're at a point where we're going to start taking the genes and trying to come up with innovative therapies, because it's probably going to be like much of the rest of medicine. One drug doesn't fit all, and we're probably going to be tailoring it to patients. So I made this point already. This is a worldwide problem. I spend quite a fair amount of time in the Dominican Republic, not for fun, but for a project down there, and I'll show you some data from that project. And we're just about to launch a study of 10,000 individuals in Mexico, and another large population that's heavily hit by this disease. But worldwide, there are a lot of people with Alzheimer's disease. And there's no effective treatment. So how did this come about? Well, we started what we call the Alzheimer's Disease Genetics Consortium, and that started in about 2002. And that was a direct pivot from this study. So in 2002, they brought people who were doing individual studies on Alzheimer's disease together. And we were not making progress. The only gene that had been identified was a polymorphism in a gene called apolipoprotein E. But we didn't know what it did, we didn't know how it worked, we didn't know why the risk was increased, and we also knew that that was only a small part of the puzzle. So several of us decided to start something called the Alzheimer's Disease Family Study, because at that time you needed linkage to do genetic studies. We still hadn't reached the SNP era. And so over the course of almost 15 years, I've collected about 3,000 families all over the world that have multiple cases of Alzheimer's disease. All the DNA is stored in one place, all the clinical data and all the follow-up is actually stored also, and we freely give it to any qualified investigator. You just have to send us, you have to have an IRB approval and proof that you can do the work, and we send you the DNA. It's the most widely used group of patients with Alzheimer's disease in the world. And when we first started this, people thought we were absolutely nuts for giving away all the information, and it turned out that this has been a real way to collaborate. And after that, the Alzheimer's Sequencing Project started. So the first step was creating this consortium, and we managed to pull together all 30 of the Alzheimer's centers across all of the United States. And each center worked up individuals that were cases. We collected the DNA. We collected as much clinical information we could, and we asked each site to send as many controls, healthy controls, and to prove that they were controls by doing some clinical testing. So this cohort is now about 25,000 cases and about 50,000 controls. And out of that, we decided to start the Alzheimer's Disease Sequencing Project. So this was, I'm going to show you data from the consortium. This was mostly genome-wide arrays, and then sequencing followed that. So the partners that were created was the National Alzheimer's Coordinating Center. So this is where all the data sits, and you, again, just have to simply apply to get the data. The National Cell Repository, we put all the DNA and all the cell lines in one place, easily obtainable. And then once we do the analysis, all the data, including the raw data, if you want to reanalyze it or take your cohort and mix it with our cohort, you can get that information from the National Alzheimer's Disease Storage Site, and this is the family study. And here are the lead investigators for all of these studies. So let me show you some data. The goal was here basically to try to identify targets that could eventually be manipulated into drugs. As you know, universities don't make drugs. We're not very good at it. What we do is identify targets and validate them, and then hopefully some company will pick it up and try to turn it into a drug. So this is how the consortium started. 30 centers, all the blood and DNA went to these repositories. First thing we did was a genome-wide array, and several additional genetic studies that I will tell you about. They were published, and all the data is in a central area where you can download it if you want. And mostly it's non-Hispanic whites, although Columbia, of course, contributed African Americans and Caucasians, and we're just now putting together a large Asian cohort. So here's the current status. MCI is mild cognitive impairment. It's kind of a pre-stage for Alzheimer's disease. And the first collaboration started at a meeting, and we were sitting in a meeting in Hawaii, and we were about to go on stage to present our first findings, and we realized that a group from Europe did the same thing, a woman named Julie Williams. So we decided to make a marriage right there. The two groups got together, and we put all of our data together and cloned out, did this large meta-analysis, and this is about 24 genes over the threshold, including ApoE. And these are genes that are associated with Alzheimer's disease. This is called for most of you probably know, it's a Manhattan plot. It's supposed to look like the skyline of Manhattan. This looks more like San Antonio or something like that, but not really Manhattan, but you get the idea. And, but the important point to note about these genome-wide arrays is they identify loci or regions. The assumption, of course, is that because the locus is near the SNP that's in SORL1, for example, actually the locus is finding a region that's in a linkage disequilibrium block. So there could be as many as 60 to 1,000 genes within that region. So a lot of people make inferences about these as being genes, but they're actually regions. They're not really genes. So the difficulty is finding the gene that's driving the locus. We also had a large collection of African Americans, something that had never been done before, and we were able to identify a locus near ApoE called ABCA7, and that turns out to be a very important gene. And just to give you an idea of the linkage disequilibrium block, these were two studies in Caucasians that identified a region on chromosome 19 that contained ABCA7, but it was really our African American study that identified this block, and later on I'm going to show you that we ultimately discovered a large deletion in ABCA7, which is a transporter gene that led to the association. We were also, because we connected with the Alzheimer's centers, able to look at endophenotypes. So some of you know that a spinal tap to look for amyloid beta in the spinal fluid and tau and phospho-tau is virtually diagnostic for Alzheimer's disease. And in fact, we just recently put together a series of 1,200 patients that have been studied at this institution and looked at the sensitivity and specificity. And it's about 90% sensitive and about 75% specific for the disease. So it's probably the best and most definitive test we have. And then when you use that as the phenotype, you can actually identify additional genes. And again, this is a genome-wide array. These are identifying regions. And we are very good, once we identify a region and find it, and we see what genes are in place, we are very good at looking up in books and trying to attribute whatever gene we find to the disease, whether it's truly related or not. But it's really ultimately taking the gene into the lab to make sure it's a real locus. And And for phospho-tau, serpin B1 and GLIS were interesting genes to be looked at. We also had enough autopsy material to conduct a genome-wide array, and GLANT1, which is a protein glycosylation, and another ATP binding cassette was found to be associated with a late-onset Alzheimer's disease at autopsy. And then we went back to the genes that were from that first large genome-wide array I showed you with the 30 genes and saw that actually all of them have a nominal significance. So all told, from the initial screens, and genome-wide arrays are really screens, we think, and these are the odds ratios including ABC, ApoE, and all the other genes that I mentioned before, we calculated the population attributable fraction. So we think 80% of the genetics of Alzheimer's disease are within these regions. We don't know all the genes yet, but we think that 80% of the variability in Alzheimer's disease can be explained by these genes.
That's non-early onset?
This is mostly late onset disease, yeah.
The early onset is a different genetic profile?
Well, I'm going to come back to that. Give me a second and I'll show you. It's not as clear cut as you think it is, or you might think it is. I know he does. He thinks a lot of things that no one agrees with. And so when you look at polygenic risk scores, we, again, explain, depending whether you look in families or whether you look in a large case control series, these two curves just basically say that, yes, 80 to 90% of the genetic risk can be explained by these 30 genes that we've identified. So to move on to the sequencing project, we had to find people who were very clear-cut because the sequencing, I think we all kind of thought that we'd find a few genes that had large mutations on them and that would explain everything, and that's exactly not what happened. It turns out that most of these genes contain multiple variations, and it's almost each family has their own private mutation. So that makes the job much, much more difficult. So we took the families that I'd collected, and we from the Dominican Republic and also from the Caucasians. Most of them were multi-ethnic, and I'll show you that. Some data from the Washington Heights study, Miami, the big family study that I mentioned, and some other studies. And then we did do some exome sequencing, and so I'm not sure how familiar you are with all of this. So whole exome focuses only on the coding region of the gene, so that's about 10% of the genome. The other 90% are all the things that regulate gene expression, and initially thought not to be so important. It turns out it's probably very important, but it's cheaper. And so initially, whole exome sequencing was done in about 11,000 Caucasians and some Caribbean Hispanics, some African Americans as well. And then, again, the Washington Heights study. So I need to go back in time because how did I wind up with 1,000 Dominican families? Well, it started with a study in New York City where we not only found this early onset presenilin family, but we discovered that a lot of our families were coming from the Dominican Republic, and there were multiple individuals affected. We also did a community study where we compared the rates of Alzheimer's disease in Caucasians, Dominicans, and African Americans, and Dominicans always had a much higher frequency of developing Alzheimer's disease. And we thought, well, maybe they have some sort of mixture. We actually did autopsy studies, cerebral spinal fluid studies, and it confirmed that our suspicion that the rates were higher. Then we started looking at their families, and we realized that, and we should have known this, but Dominican Republic is really an island population where there's a significant amount of inbreeding. So the average Dominican is really a second cousin to their neighbors. It's a little bit like Iceland in that regard. And there's no mass migration to the Dominican Republic. They do leave, and when they leave, they come to Washington Heights. So it was a perfect way to study these. And so we started this study in the Dominican Republic, and we identified a couple of geriatricians. Now, in the Dominican Republic, first thing, we had to understand the culture, the physician culture and the community culture. So psychiatrists and gerontologists take care of patients with Alzheimer's disease in the Dominican Republic. Neurologists do not. And families often, they do this amazing thing. If somebody in their family develops Alzheimer's disease, they usually buy an apartment or something next door, and that's where the patient stays. They really seldom put anybody in a nursing home. So no one needs psychotropic drugs to control behavior because some family member is actually taking care of the individual. It's pretty refreshing. I don't know how they finance that, but it works. So we started tracking down families, and we trained a couple of people. So everybody who works with us, they spend some time here, we train them, and then we work with them down there. So I go down about 3 or 4 times a year. And this is a typical situation where you walk in to see the family, and here these 3 individuals are affected. This brother is the oldest, and he's not affected, and he told us about his siblings. And so the custom there is if you go to a family's house, they all dress up for you to come, and you cannot start anything. You have to sit down and have coffee and have something to eat. So if you're going on one of these trips, you just forget any dietary issue that you're going to have, because if you don't have coffee and eat and socialize a bit, the study is not going to happen. And we go through the consent, and the good news is there's one institutional review for the entire country. So you just have to go through one, and they approve everything. And they usually are very happy to take whatever Columbia has approved. So it's not all easy like that, because sometimes we have to track people into small villages. This is Santiago, and this is looking down the street from the hospital. So and we usually rent a truck and drive around sometimes with a centrifuge, sometimes with dry ice to get everything done. And you can imagine parking here is about like parking downtown, going up to an apartment to see the families. But the families are really lovely people. The physicians are terrific. They're really well trained. They're very eager to do research. For the first time, one of our trainees got a grant from the government of the Dominican Republic to continue work there, which we were very proud of. And this is Dolly. She's the main project coordinator. It's a pair of sisters that we studied. Sometimes it's difficult. Our Jeep couldn't get through the mud after a big rain, so they came and brought our coordinators on horseback to the house so we could actually this is Angel. He's been with us the longest. We do things outside, a lot of testing outside. So we used those families to then go back to the genome arrays to see if we could find the driver. In other words, we had regions, but we didn't know for sure if they were the genes. So we sequenced all of the major genes that I showed you on that large array, and out of the 30 genes, only these 9 had mutations, coding mutations that we could identify. And this is an example. This is a family, looks like autosomal dominant inheritance. So everybody here, these 2 sisters and their offspring, or her offspring, have the mutation. And we screen everybody for presenilin and for ApoE, so we know what that status is. And these are unaffected individuals, and you can see the ages. So we've seen this a couple of times, and we just now have enough families to look at triads. So usually for a late onset disease, you can't find triads because the parents are old and the children haven't got the disease yet. But since we started, seems like decades ago, we now have offspring of parents who were both affected, and we now look at their children. And there's about a 20-year difference. So if you have 2 affected parents, at least in this cohort of families, then the average age of onset in the offspring is about 20 years earlier. And we're trying to look at which genes are being passed that way, and they're mostly homozygous.
Is that an anticipation effect?
Well, it's partly anticipation, sure, but not all clinical anticipation. So, but we are following them, so they may be getting to us a bit earlier. But these are the mutations, and I'll spend a little time discussing these in a moment. Now, Jeff, you asked a question about early onset disease. So we had originally thought that if you were, and again, it was sort of an arbitrary thing, and it was based really on the fact that when we originally started genetic studies, you needed families because it was based on linkage analysis. So you had to see transmission. So you needed 2 or 3 generations to really be sure. So the first 3 genes, APP, amyloid precursor protein, presenilin 1, and presenilin 2, were all identified in family studies first and then cloned. So we decided that some of these families looked kind of like they could be Mendelian. So we decided to sequence these genes. And so this is about half of the families. These are Caucasian families that we collected all over the U.S. and found early onset mutations in people with late onset disease. They're rare, but we found them. And then we decided to look in the Dominican families, again, late onset families, and again found almost the same frequency. What was surprising to us is this. So this is granulin, which is, and this is tau. And mutations in granulin and tau are generally associated with something called frontotemporal dementia. It's a very different type of dementia. It's a behavioral dementia. And we went back to the drawing board to say, well, did these people have any kind of behavioral phenomenon? And they didn't. So I took it upon myself to track down all of the autopsy material we had, and lo and behold, the patients have a combination of frontotemporal dementia and Alzheimer's disease. So they do have plaque-entangled disease that we see in Alzheimer's disease, but they also have FTD. So this now, we've seen many, many more cases like this. We've yet to see a patient with Alzheimer's disease only at autopsy who has a granulin mutation or a tau mutation. So that means that so this means that some people with frontotemporal dementia have a phenotype that looks like Alzheimer's disease. And we've tried to retrospectively see if we could refit it, and maybe we missed something, and, you know, it's like sort of the Manhattan view. You know, if you stand in the bathroom and look on the sink, you can see the river. We couldn't imagine any behavioral problems in these individuals. The second thing was we started seeing more families with SORL1 mutations. This is a gene we identified in 2007, and we knew a lot more about the mutations because we were able to take them into the lab now, and here are the mutations. This is A beta 40, A beta 42. These are increased in cell culture, and this is the soluble APP. So we had a biological connection. So most of it centered around amyloid pathology because that was the key deposition in the brain. But then things like this started to show up. So here is in all of the families and case control put together, because we can now do meta-analyses, coding variants in PLG2, ABI, and TRIM2. And these genes are highly expressed, not in neurons or astrocytes, but in microglia, suggesting that there's immune-related protein-protein interactions. And this has become more of the most frequent genes seen in Alzheimer's disease. And there's a little bit about, they're mostly involved in inflammation, and TRIM2 binds with tyro-BP, which then influences or activates immune cells, triggering an inflammatory response. But you can see the number of cohorts that were put together. So what we see in this now are very rare mutations in lots of different genes. So this, for example, SORL1 now has familial late onset Alzheimer's disease, familial early onset Alzheimer's disease, sporadic early onset disease, and sporadic late onset disease. And there are about 30 different highly damaging mutations. What's interesting here this is a string network. These genes are all part of a string, and there was another gene, that CD33, that sort of just missed the border of association in the large study, but it's part of this string, and it's also expressed in microglia, and I'll come back to that towards the end of the talk. We've now looked at families. This is only Dominican families, identified new genes, and the most important one is genes as a mutation in ACAP9, which is a scaffolding protein. It's several protein kinases and phosphatases involved in the Golgi. But we had also identified earlier 2 different mutations in the same gene in African American families. And as you know, Caribbean Hispanics are have about 40% admixture of African ancestry. And these are the families. So the largest family to date we have is 61 members of a family, of which 39 were affected. This was a large, so it takes us about 2 to 3 years to put together the family pedigree. This is the case control analysis. Again, ABCA7, ApoE, new sets of genes are arising. TRIM2 is a very strong player. And this is loss of function mutations. Again, ABCA7. Here's the ABCA7. I mentioned that earlier. So in the Caucasian studies, I mentioned these 2 SNPs. This is the African Americans variant. And then almost simultaneously, our colleagues in Miami found this deletion in 2 families of African American descent, and we have 2 families of Caribbean Hispanic descent who carry the same 44 base pair deletion in ABCA7, this transporter gene. We also, using this population, we were able to identify 10 missense mutations in 12 unrelated families, and it was highly significant. Now, what's interesting about this is the activator protein for the CREB binding protein. So CREB, very important in memory, these 12 mutations were in this gene called SRCAP. So SRCAP is associated with truncating mutations in the terminal exon. And these terminal exon, if it's this truncating mutation doesn't make the holoprotein. And what children develop is something called floating harbor syndrome. And it's the syndrome in which they're born with, they have a speech delay. They don't start speaking until they're 4 or 5, and they have some skeletal abnormalities. And the SRCAP, the CREB binding, the activator for the CREB binding protein is where the mutation is located. And Rubinstein-Taybi disease is a similar syndrome, similar characteristics, but the mutation's in the CREB binding protein. So this is the activator of the CREB binding protein. We have seen missense mutations. The protein is intact, but these missense mutations alter the function. And this just shows association with pathology and things like that.
Richard, can I just interject 2 questions? Can you say a little bit as you go through this about the biology and how the plausibility or lack thereof?
I'm going to get to that, Jeff.
And the other thing is, where does retromer fit in? Because I know that was another.
Well, Sorrell's one is one of them.
I see. So
Yes.
it's a class of genes that
Yes.
refers retromer.
Right. And this whole exome study in 20,000 people identified, again, Sorrel1 as the primary gene involved. And again, in early onset, this just shows more Sorrel1 mutations. And this is another retromer gene showing impaired trafficking in a small set of families. So the current status of genetic discovery is that from approximately 30 loci from genome-wide arrays, 9 genes carry coding mutations, either missense or loss of function, and that can explain it. And I've listed those genes here. ABCA7, BIN1, CDP2, clusterin are all part of the recycling or retromer function along with SOR1 that Jeff just mentioned. CR1, EPHA1, MS4 cluster, and PCOM are involved in either innate immunity or in amyloid processing. There have been novel variants that were not seen in genome-wide arrays because they're really pretty rare, and most of those are in these inflammatory pathways, TREM2, PLG21. SRCIP sort of stands out as a bit different than the others. So this caused us to start looking outside of the coding region in the epigenomic region, and so far, variants in the epigenome and some of these genes, it's basically in the same genes, some of the same genes that we identified earlier, SORA1, ABCA1, BIN1. And then, of course, there are some in genes not previously implicated, this list here. SERPIN1 is very interesting because we just started a large proteomic study that's genetically driven, and in the proteome, from cerebral spinal fluid, it turns out that SERPIN1 is one of the major proteins that seems to be overexpressed. Structural variation, large deletions and insertions have been studied. ABCA7, I showed you one, and CASP and TREM2 also have potential insertions and deletions. So I'm going to just tell you a little bit more about the complexity of Alzheimer's disease. So typically, we rely on pathology, and we see typical plaques and tangles. It turns out that only about 20 to 30% of people have only that in their brains when they die. So what else do they have? Well, they have about another third have Lewy bodies, which are intranuclear inclusions of alpha-synuclein, and they have something called Lewy neurites. But the predominant extra phenotypic thing is vascular disease. And so there's been a resurgence of interest in cerebrovascular lesions, and it's taking, we have large vessel disease, you have a large vessel stroke, you have an increased risk of developing Alzheimer's disease. Small vessel disease, seen on MRI like lacunes. And the infarctions sometimes are associated with amyloid angiopathy, and we get microhemorrhages, and this is all we're becoming aware of it because of the use of MRI. Perivascular tau and amyloid angiopathy, and it's thought to cause a hypoperfusion. So we began studying this using MRI, and Adam Brickman developed a method to do this and to give everybody a risk score based on how much of their brain had white matter hyperintensities or brain infarcts using various techniques for imaging. We validated that at postmortem looking at the MRI score that we created against tangle pathology, atrophy, infarcts, and temporal lobe pathology. And then we looked at it another way by looking at the CSF, the cerebral spinal fluid biomarkers. And then we did the first genome-wide array to look at how these things lined up with the genes that we already knew about, and that was interesting. Then we decided to go into an unbiased approach by giving everybody a cardiovascular score as a modifier and repeating the genome-wide array. And we have a new gene that we're interested in, methionine sulfoxidase, which is involved in protein repair and metabolism. So we think this may be a way of getting a handle on which genes are interacting with vascular risk factors to cause Alzheimer's disease.
Is the cardiovascular score just based on the neuropathology, or is it based on clinical diagnosis?
Clinical. It's clinical. It's a combination of clinical and radiological data. And so just to give you a picture of the current profile for Alzheimer's disease, a bunch of genes. Some of them we know, these are Mendelian, these are definitive causal mutations. These are getting there. SORA1 and ApoE are probably the most important. TREM2 also important. These have mutations, but these are extraordinarily rare mutations, less than 1%. These are still what we consider risk loci, so we know there's something in the region, but we haven't identified what the gene is. It may be epigenomic, it may be structural, it could even not be the gene that we think it is, it could be something nearby. So that needs to be worked out. The implications are the genetic variation directly or indirectly leads to Alzheimer's disease. And we think that symptoms and signs may be similar, but the pathological and etiological differences probably relate to genetic variation and some environmental interaction. Cerebral vascular disease, we think, is a trigger in susceptible people. And I think if we're going to have any success in developing treatments or preventive strategies, we have to understand the the genetic influence is a bit better. And eventually treatment will be individualized. So what are we doing with all this? Well, I'm going to walk you through what we're starting at this very moment. We had a large gift from a family just recently to set up what we call the neurotherapeutic pipeline. And the idea here is to take all the genetic data that we've ascertained over the years. We've used bioinformatics to annotate it, and we're now taking tissues from these people, mostly from brain tissue, but also from blood, whole blood, and from cerebrospinal fluid, and looking at RNA-seq, both on a cellular level and in bulk, and doing blood and CSF studies to create what we call EQTLs, or quantitative trait loci, that depict the protein that is expressed. And gene variation will then be prioritized on a multiplex screen. And I have to credit one of our graduate students, because we were kind of doing this one gene at a time, and he knew about the 30 genes, and he said to us very humbly, he said, "Dr. Mayeux, how are you going to solve this problem one gene at a time? You're never going to do it. You need to scale up, mister." And I thought, it's a really bright young man. So I said, you're right. We have to do something a bit better. So we've tried to scale up. And one of the ways we're doing this, so now any gene that, this is Andrew Spruill, so any gene that we are convinced shows up with definitive mutations, we're creating, using CRISPR, we're creating iPS cells, and we give them to anybody who wants them. Anybody wants a mutation in an iPS cell for a gene that we've done, it's available. Of course, we're doing our own studies. And then we recruited Phil DeJager from the Brigham, and he is establishing cellular specificity. So he's got now techniques to take the iPS cell. We can derive neurons and astrocytes like everybody else, and we're also making these cortical spheroids. But because the microglia are so important, we're actually now, he has a pathway to create microglial cells with and without these mutations. And then we can look at the RNA profile of the cell type and begin to conduct some pharmacological studies. Karen Duff, who's a member of this department, and Andy Teich, who's a pathologist, are both doing assembling protein interactions and testing genes in these protein molecules to see who they interact with. And the final phase is really to screen compounds to see if we can rescue cells. We're also taking cells from patients to see if we can do the same sort of thing. And Betsy Bradshaw, who came with Phil, has really done an amazing amount of work. So just to make a long story short, there's one gene I mentioned earlier, CD33, which is an inflammatory gene. So she's taken it through this pathway, and we hope to get a lot more genes through this pathway. And she found out that through the last step, the protein-protein interactions, that CD33 is not the driver. CD33 interacts with another gene called CD45, and that turns out to be the key player. And she's now working with she and Don Landry have tested compounds, and they're now working with a company to take over this to see if they can develop a therapeutic approach. So our hope is to get all of these, over the next few years, to get most of these things through this pipeline. If you're interested, all of the data is available. We keep upgrading it, so as more reference databases come out, we upgrade the sequence data. We encourage people to use the data. You can submit an application for access. You just have to have an IRB approval. It's very easy to get the data. These are my collaborators. These are Jerry Fishback, Peggy Vance, Lisan Wang, Jonathan Haynes, Lindsay Farrar, and Tatiana Faroud. She runs the cell repository. These are my international collaborators. Most of you know Rafi Lantigua. Martin Medrano is from the Dominican Republic. Yvonne Jimenez is in Puerto Rico. This is my team. Neha did the work on the cerebral vascular disease, and that's Dolly. David Goldstein really helped us get the community sequence. Peter Hislop and Katerina Rogeva are still collaborators of mine in Canada. And I get funding from the National Institute of Aging. So thank you very much.
It's on? Oh, great. Thank you so much. I'm Melissa Arbuckle. I'm the Vice Chair of Education, and Jeff had to step out early, so I'll help facilitate the discussion. Do we have questions? Yes. right here. Yeah.
I was interested in the last question about lead compounds. How good is the data for that? We had a patient from Greece where they were told that the lead was an important issue. And then we had a person here who studies different forms of lead and how it's bound to different proteins. So I'm wondering what good is the evidence for lead? And is this spa in Greece everybody goes to, and then they say, well, it's the lead. It's not a good spa because the lead gets in the system. And then our guy here who did the lead evaluations said, it's not lead. So that's a specific question.
I'm not sure I understand your question. Is lead a major cause of Alzheimer's disease?
Yeah.
No.
But you mentioned it in the last slide.
Was it what?
You mentioned it in the last slide about lead.
I did?
Yeah. The next to last slide was about lead. Just before that.
Lead?
Before that, I guess. Lead, target validation for lead
Oh,
compounds.
no, lead.
Oh, that's
Those are lead, it's misspelled. They're lead compounds.
The lead.
Okay. Yes.
Yeah, go ahead.
Just a clarification. I was always under the impression that Lewy body dementia was a very different animal than Alzheimer's, but I understood you were lumping them together as being...
Well, there's three kinds of Lewy body disease, right? So there's Lewy bodies that are, and the best review for this is by Kosaka, a Japanese pathologist who did the original work. First of all, everything changed with the identification of alpha-synuclein. So in the old days, it was H&E, and you had to be lucky if you saw Lewy bodies. And Lisa Forno in California was a fabulous pathologist who did all the initial work showing that it was scattered in normal aging and clustered in some people. And then when ubiquitin staining came available, Jim Goldman and I described about 10 patients who had looked like they had Alzheimer's disease, but in fact what they had was Lewy body dementia because they had no Alzheimer pathology. And Kosaka then followed us with alpha-synuclein staining and went back and defined three different syndromes. So if the intranuclear inclusions are limited to the midbrain, it usually causes traditional Parkinson's disease with nigral degeneration and intracellular Lewy bodies. There's a transitional form which is in the midbrain and in the limbic area, and that can be Parkinsonism with some behavioral abnormalities, not so much cognitive defects, but they can have behavioral impulse disorders, very rarely very formed hallucinations. And then there's the latter stage, or the latter form, the last form, which is cortical Lewy bodies. And cortical Lewy bodies take the form, they're not all intracellular like they are in Parkinson's disease. They can be Lewy neurites, they can be scattered, and those are associated with REM sleep disorder and very vivid visual hallucinations. The best one I can remember is a geologist from New Jersey used to come up to see me, and he also had an office in Midtown. And he said that when he was riding the train, the PATH train, that when we pulled into the station, the chairs would sort of collapse together like an accordion, and then when the train left the station, they would re-expand. And he didn't tell his wife or anything because he thought his wife would think he's nuts. And so now from many stories, and I just met Robin Williams' wife, who described a lot of these similar kind of strange behaviors that he had during his life. So there are three different forms. So in the Alzheimer patients who have hallucinations, they have mixed pathology. They have both Alzheimer pathology and they have Lewy neurites and some Lewy bodies. And Karen Martyr in our department, along with Larry Honig, have described a series of patients with Alzheimer's disease confirmed at postmortem who had Lewy neurites and Lewy bodies in the cortex, but not in the brainstem, and no Parkinsonism. So there's three different forms.
There's another question over here. Yeah.
Richard, this is wonderful for some of people. Just introduction, but description of where the genetics lie currently, and I'm just trying to square this a little bit with most of what I learned about this stuff is from the newspapers, where people are talking about the problem in identifying etiology is that you have to go back earlier and earlier and earlier in life. Well, that wouldn't be the case with genetics necessarily, would it? So that's a different way to slice and dice all this. Anything you can say to just intersect these two views, please?
You're right. I mean, the one thing in epidemiological studies, there was a problem, and it was pointed out. We all kind of knew it, but didn't really think about it much, and that is that there's probably a long period of time where you accumulate plaque and tangles, and before you start to display symptoms. And in fact, there's something called the Diane study, which Columbia is a part of, the dominantly inherited neuroimaging network, I think, something like that, in which they're taking people who carry mutations but aren't affected yet. They carry early onset mutations, studied the family members, and there's evidence of biological changes 10 to 15 years before the average age of onset in their families. And we've seen this in the couple of studies looking at pathology of healthy people that we follow. In the Washington Heights study, for example, it's not unusual for us to have studied somebody up until a year before they die. They're perfectly normal. And we look at their pathology and they have Alzheimer pathology. So I think you can have the disease, and whether that changes your behavior or changes your diet. So you can imagine if you're doing epidemiological studies asking about risk factors, and their behavior may have changed during the, you assume they're healthy because they're passing the test, but you don't have definitive evidence. So what's changed now, and there's arguments for both sides. Some people think that looking at RNA-seq and biological material at death is looking at the end stage of the disease, and that may not be so informative. But if you go back earlier, what are you going to look at? So the only avenue we have is either cerebral spinal fluid or whole blood. So there's problems looking at whole blood unless you correlate that with brain for RNA or transcriptomics. So CSF may be our only way to do that. There's not much RNA in CSF. There's some, but not much. It's hard to get different cell types in CSF. But we can do proteomics there, and that probably will be a good avenue. And metabolomics may also be helpful if we can sort out what's exogenous, what metabolomics are related to what you ate last night, and what is really endogenous to your genetic makeup. So lots of work to still do.
Alex?
So,
I'm sorry, Alex had a question. Oh, sorry, you're next.
so out of your 30 genes and the 9 genes, you actually had a remarkable amount of them were involved in inflammatory cascades.
Yes.
And so I vaguely remember, I think back in med school, there was some stuff coming out about NSAIDs and suppressing immune responses. I'm wondering what became of it and whether it's worth revisiting in lieu of the data.
Well, that's you have a good memory. NSAIDs and all of it, they tried multiple trials, but it may not be the right avenue. What we're seeing are activation of microglial cells. So Phil Dieger, who we recruited from the Brigham, is really doing cell-specific work in brain tissue, both here and from the Rush study, and trying to map out what activates these. So in the pathological studies, when they look at the microglia and the activation, again, you're looking at the end of the disease. So is that activation a result of the disease, or is it really truly causal? So one thing that, I mean, my hypothesis has been that it's the microvascular component that triggers the inflammatory response and activates these cells. So Phil and I have big disagreements about that and how to work that out, but that would be one scenario that would explain both.
Also, I mean, the genetics would certainly argue that it's not the result of the disease.
Right. But you need downstream effects. So my suspicion is out of the 30 or 40 genes, I suspect some of them we're never going to figure out why it's associated. It's probably a spurious association. I mean, you're mixing a lot of material here. But the ones where we can take them into the lab and work out the biology, those are the ones I'm going to believe. But I can't go to a lab. I mean, I'm not a lab person, so I can't go to the lab and say, hey, I have 30 genes. Can you work these out? I'll be back tomorrow. They don't work that way. So they want proof before they invest a significant amount of time and effort and money in trying to figure out the biology. So with the advent of proteomics, transcriptomics, methylation studies, we can sort of work out a lot of the pathways and guide them, and then they'll be able to work with us a lot closer.
Okay.
Richard, thank you very much for a wonderful overview. Can you say something how you, on the basis of this, what are the implications for therapy and what you dream of? What might be, in the future therapeutic?
Yeah. I think this is my dream. So it happened. I met Phil I was on NIH Council, and Phil gave a lecture, and he was talking about how he was taking cells and trying to really work towards a target, developing targets that some pharma could pick up and exploit. So it occurred to me, I had one of these life-changing events, and I say, well, I've been doing this for a long time, and I found all these genes, but they're not helping anybody, and I had at a lay conference with convincing people to give us their blood and let us study them. The guy and the man in the back said, well, if I have all these SNPs, what does it mean, Doc? I couldn't answer the question. I said, well, it could mean a lot, but I couldn't really directly answer the question. So the goal here is to now go from here to get into this pathway. And I'm assuming that some of them will work and some of them won't. I'm assuming that some things will get to here and we won't see much change in the cell, or we won't be able to work out anything. And I think when we get down in here, there's more hope. I can just tell you, for example, on the protein-protein interactions, so SORL1 has been a gene we've been working on since 2007, trying to work. So no doubt it's a real gene, no doubt about it. So it turns out that one of the mechanisms for SORL1 that we proposed was that somehow mutations would prevent SORL1's function of taking the amyloid precursor protein through the recycling, Golgi, et cetera. But if it didn't go through that pathway, it would go through the base pathway and form A beta fragments, which were bad. So it turns out that SORL1 interacts with a completely different protein called RTN4. Now, we would never have gotten to that point without Andy Tyke and a young man named Fallick who works in the lab who mentioned this. He's got to work out the biology and everything else about how this works, but it turns out that RTN4 probably pulls the gene into the base pathway, or pulls the protein into the base pathway. So these kind of mechanisms I think we have to work out. And my guess is at the end of the day, your therapy, if we ever get to new therapies, would be based on what your genetic profile is. So I think that the current use of amyloid antibodies is going to be extremely limited. There'll be a few people that will respond because they either overproduce or can't clear amyloid beta. But this is what accumulates at the end of the disease. We have to be way back here before anything starts. And I don't think the current therapies are designed that way.
Stephen?
Yeah. I'd like you to say a couple words about your unaffected controls and what they're like. And I'm just wondering if another strategy might be to look at the other end of the spectrum, which is to take a look at the extreme old aging ones, 100 or so, who are unaffected. And have you or anyone studied them? And is there something that's protective rather than put someone at risk?
Yes, that's a very good thought. Tom Pearls, who collected these centenarians, and Neil Barzilai at Albert Einstein's collected these 100-year-old people. And so I asked if I could review their data. And so they just do it by history. So if you test the people, many of them are demented, but they're 100 years old. So physically they're living a long time, but they're demented. So they have the same frequency of dementia as you would anticipate. So there wasn't any saving there. What we're doing is something slightly different. So there's I didn't bring the slides, but there's 2 studies. So it turns out that in a small number, we have about 40 people who are in about 15 families who are homozygote for ApoE4. So their risk for Alzheimer's disease is tenfold higher than anybody else's who doesn't have it 4. So they somehow have survived into their 70s and 80s without evidence of disease. So we've sequenced them. We've got some genes that may be modifying their effect. So I said, well, that's probably not good enough because we don't have autopsy data. So we did cerebral spinal fluid in studies in the people. And so a small number of them have a profile that looks like they should have Alzheimer's disease. They meet the CSF criteria. The rest do not. They have normal CSF. So they're somehow escaping. So that's the group I think I'm going to put my money on. Another study is a person named Joe Lee that works in our group. We discovered in, I think it was about 2000, a group of people in Puerto Rico who have a presenilin mutation. And the presenilin should cause early onset Alzheimer's disease. So what we discovered was that there's a high degree of variation in these families. So some people in the family, there can be as much as a 40-year difference between when you get the disease. Not if you're definitely going to get the disease at some point. But some people in the same family got the disease at age 25 or 30, and others didn't get it till 70. So Joe now has taken that, Joe Lee has taken that, he sequenced those families. He's now got a new grant to actually test the effect of the mutations or the genes that he's identified to see how they're explaining that difference in age of onset. I think that's how we're going to get the answer. I don't think studying people that manage to survive, even though they're important, I'm not saying they're not important, I don't know that there's anything there. I think we have to go back to this drawing board.
Deborah?
So interesting, great to see you. You know, you said there's no cure, no good way to treat at this point. But certainly lots of people who have MCI and Alzheimer's go to doctors and get medicines. And there are new things that people are trying. I recently read about nicotine patches that are being studied. I think Mary Sano is involved in that study. Should older people who have dementing processes not be taking these medicines? I mean, in other words, like if you go to sort of an average neurologist or GP, lots of people are going to get medicines.
Well, they're the only approved medications that we have, so...
But what about that? I mean, is that...
I mean, they're not harmful. So if they're approved, that means they've gone through some sort of a randomized trial. I don't discourage people from doing this. Most of the clinical trials that are experimental, people are allowed to stay on the medications that they've been prescribed. So, at this point, I don't believe anything that's not gone through a randomized trial. There's a way now you can bypass it. There's ads on TV for eating dried jellyfish or something like that. And that was first attempted as a randomized trial and failed. And so now it's direct to consumer because it's a food product. So if it doesn't go through a randomized trial, I just don't think it should be.
Additional questions? Yeah, Simon.
Do you think it's an issue of time that, in terms of these amyloid antibodies and these things that attempt to clear the end-stage pathology, that it's really an issue of not acting early enough? Because I think a lot of polygenetic diseases, in the rest of medicine, we treat the end state in hypertension or COPD or asthma, and we don't have as much difficulty getting a treatment response. It seems only in psychiatry and neurology, when we're treating the final pathway, do we really just have these really bad...
Well, they're testing that hypothesis, right? So in this study I mentioned earlier, the Diane study, so they're doing a preventive trial it's about 18 centers across the world that are all participating in it. They're giving them these amyloid anti-amyloid, or these amyloid antibody therapies in people who are carriers of these mutations, but they are currently asymptomatic. And also there's a study in Colombia and Kindred. There's a pre-Snellen mutation that's slightly different from the one we have in Puerto Rico. And they're doing a randomized trial in that cohort, again, of the same types of therapies. But those trials have been going on for a while, and they haven't reported anything yet.
Rachel?
I was just wondering if you could comment on some of the recent research suggesting that herpes virus might also be involved with Alzheimer's disease and how that might relate to some of the work you're doing with these inflammatory pathways.
Yeah, that was an interesting paper. If you go back and read the paper very carefully, a lot of viral fragments were identified. They latched onto herpes, and it's baffling to me why they picked that one, because there were equal viral fragments in for other viruses in that, and most of us have had a lot of skepticism about that. It's not been replicated, so I mean it comes from a reputable group, so you have to, you can't just write it off. But the methods were a little bit strange, and they came out of. Now the other thing is that old brains collect a lot of stuff. So, and herpes virus lives in our nervous system. So it could conceivably be an artifact, but I think, jury is out. If somebody replicates it, then it's probably believable. I know there are trials now of vanciclovir and other anti-viral agents, so we'll see. My guess is it's going to wind up being like, when the anti-inflammatory story, as the gentleman brought up before, they did a trial of prednisone, and it was a miserable failure. And some of our best ideas don't seem to work out, like aspirin.
Dr. Forrest?
So there's so many causes, and it all gets built up. Presumably there's a removal system that's going on with the lysosomes. What about jacking that up, whatever the cause, or somehow figuring out what the normal removal way is? And how do antibodies, how does that remove, or does it remove the stuff? And also, is the stuff the cause, or is it just another?
Well, it's a very good. That's the question that they're trying to resolve. They can clear the amyloid plaques with the antibody therapies, but people don't get better. And people think if you give it earlier, maybe it would. But my guess is the amyloid plaque is sort of the end stage of something, and that the cells are probably sick for a long time before that. And in the scenario that I like to think about is that you first lose synapses, and there's pretty good data to that, or connections between nerve cells, followed by, once the cell loses a synapse, then proteins don't act, don't behave, they misfold, they cluster, they aggregate, cells kick the proteins out into the extracellular space, and so plaques could actually be protective. One could argue they maybe are protective in some way, but clearing them, they can certainly clear them, but they doesn't really affect the patient at the end of the day.
Another question? Yeah.
You mentioned some of the studies about different ethnic groups and worldwide incidence. Are there any areas in the world where they used to claim in Japan that they had a very low incidence? Is that validated, or are there any other studies?
Actually, Japan has pretty high frequency.
That's
They
what I
have
thought, yeah.
They have a lot more vascular disease,
Yeah.
but their incidence rate, it's an aging population. It's actually aging much more rapidly than the U.S. population, even though we're aging pretty rapidly as well. They used to say that you didn't have Alzheimer's disease in Africa, but of course people don't live to 80. But any country that has a population that's getting into the 60s and 70s, the frequency is about the same. The incidence rates are about the same.
Great, well, thank you, and let's continue the discussion upstairs at lunch.